Okay, so we're going to pick up right where we left off. In case you didn't, for some reason, got the names backwards or something, this is not the beginning of the tutorial. This is actually the second half. The first half is labeled Biology, Genes, or DNA something tutorial part 1, and this is part 2. And if you are doing this right after you did part 1, for the love of God, take a break. There's a lot of material here. Okay, so far we've talked about the structure of DNA, how DNA copies itself, and how DNA makes proteins. If you didn't do this activity, you really need to, probably even before you start part 2, because it talks an awful lot about the relationship between DNA and proteins and how we make proteins and what proteins are made of, and most of that is in the tutorial. I'm sorry, in the video, not in the tutorial. So if you're not watching the video, you are really making a mistake. But if you're watching this one, you probably not watch that one. So we're going to move on, and now we're going to start talking about a guy named Gregor Mendel. Gregor Mendel was a monk about 150 years ago, maybe even longer. You know, it was in the mid 1800s, so it was in the 1850s. And he lived in Austria, which is in Europe, and he was a very quiet man, very intelligent, but kind of weird. You know, today probably just be one of those really geeky people that walk around, you know, looking at calculators all day. But Mendel had some pretty important characteristics about him that I want to talk about. He was the right man for the right job at the right time, and he figured out how we inherit our traits. See, up until then, we really didn't know. We didn't know how they were getting there. We didn't know the rules of it. Mendel was able to study the math of trait inheritance and describe it very well. And he's one of the most famous scientists of all time. And there's a lot of most famous scientists of all time associated with genetics, such as Watson and Crick. You saw them in the last video. And here's Gregor Mendel. The thing about Gregor Mendel is he died. Nobody even knew who he was. It was 20, 30, 40 years after he died that somebody found his paperwork and really started using it. So even in the 1850s, when Mendel was figuring out the mathematics of inheritance, we didn't know about it, and it wasn't discovered until the 1920s, well, 1900s, before we started studying it and really applying it. So this idea of genetic manipulation, not manipulation, well, yeah, but this idea about genes and understanding DNA, you know, it's only about 100 years old. And it may seem like a long time to you, but it's really not. And so we're talking about a relatively brand new science. Compared to other sciences, this is one of the newest sciences around. And so it's kind of amazing how much we've learned in the last, I would say, 100 years, right? Now take a look at this gentleman here and his son. You can see some very similar features that they share. They both have similar skin color, similar body shape. They both have the same color eyes and eyebrows. They've share some more obvious characteristics. They both have ears that are in the same place, you know, and they're not donkey ears or rabbit ears. So why is that? Why does this person right here have the DNA that tells his ear to grow like that, instead of a donkey or a rabbit or, I don't know, whatever else has ears? And then why does it then get passed along to his offspring, who has a similar ear shape? And by the way, ears are funny. They're like fingerprints. No one has the exact same ear structure, but you know what I'm saying, right? I mean, what is it about, how did he pass that information along? Okay, and how is that expressed? And so we're going to be studying that with Gregor Mendel. Now, a little bit more about Mendel. This is a picture of Gregor Mendel, one of the few that we have, and he is called the father of genetics because he was the first guy to really systematically study it mathematically, and math is the key to this. And so Mendel, you know, worked his way through school. Well, okay, he was born on a farm, and he was very smart, but he was also kind of a wimp. And I don't say that in a bad way. I'm just saying that he didn't take to working the family farm. You know, that was a lot of physical labor, strapping the horse to the plow and plowing all day. And he had little brothers and stuff, and his dad worked and you know, ran the farm. But they noticed that he was kind of a weak kind of a person, but he was very smart. And so some people got together and decided that they would send him to a monastery, and he loved it. A monastery is a very quiet place, and he was able to just be quiet all day, go do work. They put him working in the garden, and he came back in his room, and he had access to the library at the monastery, which is a weird thing because that's not a common thing in the 1800s, in the 1850s or 60s. So he had access to all this information, and it was just the best thing ever. It was quiet. He did his work. He studied. He learned, and it was great. And he showed a lot of aptitude, and so much so that the monastery paid to send him to the university in Vienna. And now that was a rare thing. Today, lots of people go to college, but back then it was really rare to go to college. So they sent him to college, and they said, look, we're going to pay for you to go to college. We want you to get a degree in whatever, and he decided math. And then he would come back to the monastery and learn, use his skills to make the monastery better. And he thought it was a great idea. He absolutely planned, and he did. He came back and he lived the rest of his life in the monastery. Eventually became in charge of the monastery. But when all this happened, he was studying. He started noticing things about pea plants, and we're going to talk a lot about pea plants. But he studied pea plants before he went. They grew all kinds of stuff, but peas, and we're talking about like just green peas. Like, you ever had a frozen bag of green peas? This is what we're talking about. And he noticed some very interesting things about them, how they had certain traits, and how the next generation had those certain traits. And he learned how to grow them and how to control their pollination, instead of letting it happen randomly. And then he went to college and studied math. One of the things he studied in math was a brand new type of math called statistics. And he came back after he got his degree, and he went right back to working in the garden, and he learned an awful lot. And he applied his understanding of math, especially statistics, to the peas. Okay, and so he also knew that he had to control the pollination. He couldn't just let the pollination happen randomly, but he wanted to study how the traits moved from the parent plant to the offspring plant. And by the way, this is long before we knew about what DNA was. This was long before we knew what chromosomes were. We could see chromosomes in the microscopes, but we had no idea what they were, and we certainly didn't know they were involved with inheritance. And so for Mendel, this was all mathematical. He didn't understand the mechanics inside of the cells as to how this worked. He just understood the math. But he knew that he had to learn how to control the pollination, so he did it in a greenhouse where there was no insects that could come in there and do pollination for him. He worked in a greenhouse. It was actually very small, about the size of a garden shed. But he would go in there and he would take meticulous notes and carefully count or keep track of what parent plant reproduced with what parent plant to produce an offspring plant. And he learned how to cross-pollinate and self-pollinate, which are very important. Self-pollination allowed him to get a pure strain of a particular trait. Then he learned how to cross-pollinate. And by doing all that and then applying statistics to his observations, he then figured out mathematically all about what we call simple Mendelian genetics. Now, at a basic level, this is what Mendel's experiment looked like. And I want to be clear, we're studying one characteristic here. That's the color of the peas. Okay, Mendel studied seven different characteristics in these peas, but we're only looking at one. Okay, and we'll talk about what seven are in just a minute, but right now we're looking at one characteristic, and that is the color of the peas. As you can see, the ones on this parent, the peas come out with a very sharp green color, and with this parent, the peas come out with a slight yellowish color. Now, they don't really look yellow like that, but they look more like of a lime green as opposed to a true green. But Mendel noticed that peas came out either green or yellow, and that's it. You never got any other combination, green, yellow. And it was kind of funny to him. So through self-pollination, he kept breeding green producers together until he got a strain of these that always produced green offspring when bred to other green parents. And he did the same thing with yellow. He bred yellow until he got a strain of yellow plants that always produced yellow every time. Okay, and he called these the P1 or the parental generation. Now, we know today that they are genetically pure for green or pure for yellow. And then he said, okay, what happens when I cross them? Now, at the time, it was held that you were just some kind of combination. You were just some kind of a mixture between the two parents. So he would have expected, and everybody at the time would have expected, that you've gotten some kind of a yellowish green mix. But to his surprise, it came out all yellow, exactly like one of the parents. In fact, the green trait totally disappeared. This was odd to him, and he asked the question, well, if the green trait disappears, how come we still have green traits? After time, it should all be yellow. So he took what he called the F1, or the first filial generation, F-I-L-A-L, which means sibling. He took the first filial generation and bred them together and was very surprised at the outcome. He got a lot of yellow, but look there, there's the green. This told him a couple of things, but one of the main things it told him was that this green trait absolutely, positively must be hiding inside of the F1 generation. There's just no other way around it. It's there, it's hidden. And this is how he came up with his idea of having genes from both parents. Obviously this parent put a green gene in here, but it was hidden, and that's what he called it, hidden. It was covered over by the yellow, and so the yellow must just be stronger than the green in this generation, but it still shows up in this generation. And it always ended up in a 3-to-1 ratio. 75% came out yellow, 25% came out green, and he knew what a 3-to-1 ratio meant. He had seen it before. And I'm going to give you a little example. Take 2 quarters and flip them over. How many possible combinations do you get? And the answer is 4, believe it or not. If you take 2 quarters and flip, you get 4 possible combinations. Let me show you. Here's another way of looking at it from Mendel's perspective. Mendel, being a statistician, they played statistics teaches you stuff using simple stuff like flipping quarters and things. But Mendel had seen that 3-to-1 ratio before, and it came out like this. If I have 2 quarters and I flip those quarters, there are 4 possible outcomes. You may have thought there were 3, but there's 4. There's heads on one, heads on the other, heads on one, tails on the other, tails on the first, heads on the other, and then tails on both. That gives you 4 possible outcomes. Okay? And what Mendel knew was that if you classify them like this, if you say the chances of flipping 2 coins and not getting at least 1 heads is 1 to 3, or in this case the chance of flipping 2 coins and always getting at least 1 heads is 3-to-1. There's only one way to not get heads. Okay? And so Mendel knew that this was a 3-to-1 ratio. He'd seen it before, and so he knew that a 3-to-1 ratio meant the odds of having 2 events, flipping a quarter over here, flipping a quarter over here, each event having 2 possible outcomes, you can get a 3-to-1 ratio. This was really important in him understanding how inheritance is caused happens from 2 parents, because he knew he had 2 parents. And if he has a parent here, that would be this side, and a parent here, that would be this side. To get a 3-to-1 ratio, it must mean that they send 2 sets of DNA. This was revolution. This was brand new. Nobody thought about this, but he thought the only way for this to happen is if a parent here sends a full set of DNA, or has a full set of DNA but only sends 1. That would be like flipping a quarter. Now, I don't know if you remember, but we talked about your parents giving you each a set of DNA. You get 1 from your mom and 1 from your dad, so you have 2 sets of DNA. That's kind of like the heads and the tails on a quarter. But when you flip a quarter, you can't get both heads and tails. You just get one or the other. So when you reproduce and you send genetic information, you're only sending 1 of 2 possibilities. The same thing with the other parent. And that's exactly what Mendel figured by looking at the quarter flipping analogy. That's how when he saw 3-to-1, that's when he figured it out. That's when he figured that these 2 organisms inherited from, this is a brother to this one, so this is an offspring here. It inherited both traits. It inherited 1 from mom and 1 from dad, and it's in there. And just because the green isn't showing doesn't mean it's not hidden. But when it passes along traits, it passes them along with the chance of both going. So this offspring from these 2 parents got the yellow. This offspring got the yellow. This offspring got the yellow. This offspring did not get the yellow, and neither did this offspring, because it must mean that since these both have a green grandfather, that they're both hiding green, and there is a chance that they could both pass green along to one of the offspring. Now that chance is low. It's only a 25% chance, as demonstrated by getting 2 tails on the set of quarters. But it's there. One of these represents getting both yellows from both parents. One of these represents getting both green from both parents. And these 2 represent the odds of 1 parent giving a yellow and 1 parent giving a green. And so then he thought, well, how come they don't mix right here? And that's when he realized that 1 color, yellow in this case, must be dominant over the green. It was brilliant. It's a little hard to understand, but this is what made Mendel such a genius. And he performed thousands of experiments on all kinds of traits, not just the seed color, right here. Okay. He also looked at the seed shape. Some of them were very perfectly round, some of them were slightly wrinkled. He looked at the colors of the flowers. Some were white and some were purple. He looked at the form of the seed pods. Some were constricted, like they were vacuum packed, and some were inflated. He looked at the color. Some of these seed pods were yellow and some were green, kind of like the peas themselves. He looked at the plants. Some of the stems would grow, some of the flowers would grow up on the axial pod and some would grow on the tip. He just knew that was normal. And finally he looked at the size. Some of them grew tall and some of them grew short. These 7 characteristics, Mendel got very lucky because these are 7 characteristics that are what we call simple single dominant transmission. We have another word for it, but most of the traits that humans have require multiple alleles instead of just one allele. Each one of these represents an allele. So he got lucky with peas, but he also was very smart. And he performed that experiment over and over and over again. And, got the same results every time. 3 to 1, 3 to 1, 3 to 1. If you watch the video that I'm including, there's a really entertaining and well done video about Gregor Mendel. And, the actor that plays Gregor Mendel just sits and takes a whole bowl of peas and counts them out. Yellow, green, yellow, green. And, he gets like, 1,200 yellow and 400 green, which is a 3 to 1 ratio. And he does it with just over and over again. And Mendel did this to the point where he almost went crazy. But he kept meticulous notes. Another thing that Mendel was good at, keeping track, keeping good notes. And all this stuff added up to us later finding this information and going, wow, this is it. Look at what this guy figured out. And of course, Mendel was long dead by then. Keep in mind, Mendel did all this without even knowing what a chromosome was, had no idea how chromosomes behaved during prophase, metaphase, anaphase, and telophase. Again, another video that I've included, and I'm probably going to show that to you in just a minute, shows you some things about how the chromosomes move during mitosis, and that's important for Mendel's work. One thing that Mendel didn't know about chromosomes was that genes were stored on pieces of a chromosome called an allele. And this area right here, this gene, is called an allele. Okay, it's nowhere near that big in comparison, but it's made that big so that you can see. And now what he did know was that parents had 2 traits. An organism had 2 traits, one from each parent, okay? And so he labeled them using letters. It was just simple for him. Like if he were looking at color of the peas, he would put a capital Y for yellow and a lowercase y for green to help him know the difference, that yellow was, I don't know that he used the word dominant, but we're going to use the word dominant. Yellow was dominant over green. And if that's the case, then there's 3 possible parents. You got a parent that can have both dominant yellow. That would be big Y, big Y. We call that homozygous dominant. You can have a parent that had both green, little y, little y. We call that homozygous recessive. Okay? And then you can have parents that have one of each. That's heterozygous. It is analogous to getting heads, heads, tails, tails, heads, tails, and tails, heads. But I don't want you to get caught up in that right now. But I want you to know that because of Mendel's observing the 3 to 1 ratio, he started figuring all this out. Now, here's what really happens. You inherited this chromosome. You have 23 chromosomes, okay? But for each one of them, you get a pair, one from mom, one from dad. Let's just pick a number. We'll say 17. This is chromosome 17. You inherited this chromosome 17 from dad, and this chromosome 17 from mom, okay? Now, they look a lot alike. They're shaped a lot alike because, they're the same chromosome. You just got one from mom, one from dad. If from mom you got, this was dad. If from dad you got the yellow trait on that chromosome, and if from mom you got the yellow trait on that chromosome, guess what? You yellow. There's no other option, right? If you got the green trait from dad and the green trait from mom, guess what? You green. But what confused Mendel until he figured out that one just totally dominated the other was, what happens if you get a yellow from dad and a green from mom, or a green from dad and yellow from mom? Why didn't they come out mixed? And that's when he came up with his idea of dominant and recessive genes. The yellow must completely cover over the Y, and the Y just stays dormant or hidden or recessive is what he called it. Now this should be familiar to you. You've probably studied this before, but I want to be clear that you understand this. When we say dominant, we're talking about a spot on the chromosome that contains the gene, and that gene is dominant over the second option, which is the yellow, which is the recessive. Okay. Now there's almost always just 2 options, a dominant, recessive. Every once in a while we get a third option, but it's really weird, So most of the time, for every gene, you got a dominant and a recessive. It's kind of weird. Because of all this, we couldn't just say things are yellow and green, and now we have to know a little bit more about the internal workings to understand how things are inherited. So if before Mendel we didn't understand this, we just thought some are yellow, some are green, that's it. But now we understood that some are yellow, but they're yellow and different on the inside. So we came up with these terms, genotype and phenotype. Phenotype simply means what does it look like. I think phenotype sounds kind of like feel, so I think feel, does it feel fuzzy or does it feel, I don't know, smooth? Phenotype means what can you just tell just by looking at it, right? The seed pod is yellow, the seed pod is green. And if you didn't have this side right here, you would assume that these are the same. But Mendel showed us that they're not. And so on the inside, we now have to look at their genes, okay? The yellow could be dominant, or the yellow could be dominant recessive. Now the green being recessive is always the same thing. It's always recessive. There's no way to get green or any other recessive trait in this case without having it both recessive. That just can't happen, not with simple Mendelian genetics. There are ways where it happens in other, but that's like real freaky stuff, and it's so unbelievably rare we just don't even consider it right now. So just while you're learning this, just understand that the only way to get green is for it to be little y. That's it. That's the only way you can do it. You can get big Y, and big Y and still have yellow. So if you see green, you know it's little y. But if you see a yellow pea pod, you don't know. It could be big Y, or it could be big Y. And so how do we tell? And better yet, how can we predict? And that's what Mendel started trying to say, is he could predict what the offspring would look like based on the parents. And this is what really started us down the path of understanding genetics. So as a result of, it took him a while to understand that 3-to-1 ratio, and he finally figured it out because it means that each parent has 2 traits and only sends 1. They call this the law of segregation. And when producing sex cells, you only get 1 of the chromosomes from the parent to go into the new into the sex cell, right? And then those sex cells combine during fertilization to produce a whole new combination of genes. So that's how you get 2 in the new one, because you only get 1 from each parent. He called that the law of segregation. And the law of segregation was basically this: a parent has 2 genes, but they're only going to send 1 to the offspring. That's called the law of segregation. And once he figured that out, once he was able to understand that the parents have 2, and there's 2, it's a flip of the coin, which one goes to the offspring, but the parents have 2 possibilities and only send 1 to the offspring. That was called the law of segregation. They literally segregate out, or they separate out. So, you know, this, if you look at this Punnett square, which I don't know if you remember doing Punnett squares, but this is a parent right here. This parent had a dominant and a recessive, but it only sent this circle right here, or this P right here, represents the odds that this offspring got this trait from the parent. This one represents the odds that it got this trait from the parent. And that's what Mendel said by segregation. Even though the parent had 2 possible genes or alleles, it only sends 1 to the offspring. The other one doesn't go. Same thing for the other parent. So there's 4 possible outcomes. This is exactly what we did when we were flipping the quarters. We got 4 possible outcomes. This would be heads, this would be tails. So this one got heads from one parent, heads from the other. This is the odds that the parents got heads from one parent and tails on the other, heads from one parent, tails on the other, and tails from both. That's why I showed you that example. Okay. And then of course there's ratios that I want you guys to notice here real fast. Notice what we call the phenotype ratio. What does it look like? We've got 3 yellows and 1 green, so we give a 3-to-1 ratio. That's right here. The phenotype, or what it feels or what it looks like, the phenotype ratio. Now the genotype ratio is a little different. How many do we have that are both dominant? That's just going to be 1. How many do we have that are 1 dominant, 1 recessive? There's actually 2 of those right there. Okay. And then finally, how many do we have that are both recessive? Now here's the thing. You may be saying to yourself, wait a minute, I see how this is big Y, little y, but why isn't this one little y, big Y? And it's because you have to think of this in 3 dimensions. This is big, little y, big Y, but then we just flipped around like a card, and now it's big Y, little y. Okay, so I've got one that is homozygous dominant. It's got the same traits, that's homozygous, and then it's dominant because the traits are dominant. I've got one that's homozygous recessive. It's got the same traits, that's what homozygous means, but it's recessive. And then I've got two that are heterozygous, meaning they've got one of each. And this is called a Punnett square. This Punnett square only looks at one trait, and we call that monohybrid. Mono being one, hybrid meaning, well, hybrid meaning cross, but it refers to the fact there's only one gene being looked at here. And the law of segregation can best be explained by this, which is everything I just went through, okay? And so each square here represents the possible outcome. So for these two parents, both of them having a big Y and a little y, there's 4 possible outcomes. Well, there's always 4 possible outcomes because that's just like flipping the quarters. But in this case, 2 of the outcomes end up being the same. So there's really only 3 possible outcomes, right? Then that's not this ratio here, but I've got 3 possible outcomes. I have dominant, dominant, recessive, recessive, and a dominant, recessive. Those are the 3 possible outcomes. And so the question is how many of each are there? Because the parents can be different. Could this parent have been big Y, big Y? Sure. Could this one have big Y, big Y? Sure. And if you fill this out, then you would get all big Y's. So there's only one possible outcome there. Sometimes we can get 4 to 0 ratios, 3 to 1 ratios, and 2 to 2 ratios, even 1 to 3 ratios. That's how these Punnett squares work. You really should, I'm going to link a video for how to do these. You really should follow along on how to do those, because I think there's a question on the test. Okay, if I give you this parent and this parent, what are the odds that it'll get this offspring? It's not that hard if you know how to do these Punnett squares, so you should practice. And that's exactly what I'm telling you right here. This does not represent the fact that this parent reproduced with this parent and had 4 children. That's not what happens. It represents the odds. So for every single offspring, the odds are 20. In this case, with these two parents, there's a 25% chance that it's going to get a big Y, big Y, a 25% chance it's going to get a little y, and you add these two together for a 50% chance it's going to get a big Y, little y. See, we talked about this. Now, if you just pretend that's a card and you flip it over, it's big Y, little y, right? So for these two parents, every time they reproduce, whether they have one child, like 1 baby P, or 2 million baby P's, there's still only a 25% chance of getting big Y, 25 little y, and a 50 of big Y, little y. Okay? Now in these cases of P's, yeah, you know, P's reproduce a lot. They have lots of offspring, hundreds. Okay? But if this were a human and they only reproduced and made 1 child, then there you go. Same outcome. Because the rules are the same, whether you're passing along pollen or passing along sperm, the rules are the same. Okay? Well, that was only looking at one trait, in this case, color of the pod, color of the seed. But you know, we can actually predict 2 traits. And by the way, we're predicting, I don't know if I made that clear, we're predicting what are the odds that these 2 parents would produce this color offspring. We're predicting. I grew up on a cattle ranch, and to some degree I'm starting to be heavily involved with my family's operations. And it's the same thing. If I've got that bull and that heifer, what are the odds I'm going to get that kind of calf? And it's the same kind of genetics, you know, and I'm kind of impressing my father-in-law by showing him that I totally understand that. And he said, well, how do you know that? I said, I teach it every day in science. But we're looking at only one trait. What if we wanted to look at 2 traits? In this case, what if we wanted to look at the color of the pea and the shape of the pea? Instead of calling that a monohybrid cross, where we're only looking at 1 trait, we're going to look at 2. We call that a dihybrid. We can do this with cows. In fact, it's pretty easy to do, really. We can do it with horses, we can do it with dogs and cats and, you know, whatever. Because almost every sexually reproducing organism on Earth does this. They have 2 traits. They're what we call diploid cells, because they have 2 traits for every organism. For every characteristic, there's 2 traits. I know that's confusing. Okay? Now you may say to yourself, wait a minute. There's more than 2 skin colors, there's more than 2 eye colors, more than 2 hair colors. Yeah, I know. So keep that question in mind, because we're going to explain that later. That's called polygenic inheritance and multiple alleled inheritance. But believe it or not, with very few exceptions, everything has a dominant and a recessive. Okay? And I'll explain that later on, right? But look, what if could we predict 2 traits? And the answer is absolutely, using the exact same method. Probability is probability. And now we're just flipping 4 coins, but this time we're flipping 2 quarters. That's going to show us the pea color, and we're also flipping 2 nickels. That's going to show us the pea shape. And so it gets a little weird, and it gets a little hard. This is not easy. It is easy once you know how to do it, but it's not something that you can do just by looking at it. But I don't think this is going to be on the test, so I'm not teaching it to you. But one of the things we can learn from this is called the law of independent assortment. Remember how I told you that every parent passes along the traits? You get one of your traits from mom and one from dad. Well, dad has two, but he has two of each. And what Mendel learned was that just because he's passing along the gene from one of his parents to you doesn't mean you're going to get all of his dad's traits passed on to you. Let me see if I can show this to you in a graphic. Okay, so here's my attempt to explain this. This is you, okay? This is your dad over here, and this is your mom. Now the numbers on top represent the chromosomes. We number our chromosomes. We number them 1 through 23, right? And they're based on size. So the bigger ones are longer than the shorter ones, right? So as you start at number 1, you're looking at the biggest one. You go right into 23. That's the smallest one. So here's the thing. So we're looking at 2 chromosomes for your dad. Now each of these, this is chromosome number 4, and this is chromosome number 7. But remember, you got 2 of each, one from mom and one from his mom and one from his dad. So we're going all the way back to your grandparents here, right? So when he was born, he had one of your dad's chromosome number 7s and one of your mom's chromosome number 7s, and he had one of your dad's chromosome number 4s and one of your mom's chromosome number 4s, and he had one of each other one too, the 21 other chromosomes. And so he's diploid. He's got one of each chromosome, one from mom, one from dad, one from mom, that makes him diploid. Okay? Now if this one has a dominant on it and this one has a recessive on it, then he's going to show the dominant trait. If this one has a dominant and a dominant on both, then he's going to show that trait. If this one has recessive on both, then he's going to show that trait. And by the way, there's hundreds of traits along here, and we only look at one. I mean, this is how confusing and complicated it can get. We're only looking at 2 traits right now out of thousands, hundreds of thousands, right? Your mom is the same way. Your mom had grandparents. She had a dad and she had a mom. And one day your grandparents decided to have a child. We're not going to go into how that works, but the dad passed along this chromosome number 4. Your mom passed along, or your mom's mom passed along this chromosome number 4 to give your mom a full set of number 4s. Then they passed along this number 7 and this number 7 to give your mom a full set of number 7s. One came from dad, one came from mom. And so the question is right now, and this is what the law of independent assortment means, is when your mom and dad reproduce, and no, we're not going to talk about it, but yes, it did happen. When your mom and dad reproduce, you don't get both of these. You only get one of them. And that's the law. Well, that's the law of segregation. You only get one of these. One goes away. Actually, it goes to a different sperm, but that's another story. So what are the odds that you're going to get this one or this one is 50-50? The odds you're going to get this one or this one is 50-50, but the odds you're going to get any particular combination is 1 in 4, which we just talked about with the Punnett square, right? You can call this one heads and this one tails and this one heads and this one tails, and you flip 2 quarters and that's what you get. But what if dad sends me, I'm going to do some funny stuff here. Whoop, that just disappeared. Hold on, I know how to fix this. Bink, look at there. What if dad sends me his dad's number 7? Does that also mean he's going to have to send me his dad's number 4? And the answer is no. Okay? Because of the law of independent assortment. And that's what the law of independent assortment says. Just because my dad, sent me his dad's number 7, boom, doesn't mean that he has to send me his dad's number 4. He could send me his mom's number 4. That's what it means by the law of independent assortment. And your mom could do the same thing. Just because she sends me her mom's number 4 doesn't mean she has to send me her dad's number 7, or her mom's number 7. She could send me her dad's number 7. And that's how we get you. And this is chromosome number 4, one from mom, one from dad. This is chromosome number 7, one from mom, one from dad. That's called the law of independent assortment. It's very simple. Just because dad sent me his dad's number 4 doesn't mean he has to, or number 7 doesn't mean he has to send me all of his dad's DNA. They're sent independently. That's why it's called the law of independent assortment. So that happens all the time. That's why we have to allow for every possible combination. That's what the dihybrid cross does, is it shows every possible combination of the two genes from each parent. And it creates 16 possibilities instead of just 4. And it gets very complicated. If you actually had to do these and count, you would come up with a ratio that has 4 numbers in it. And by the way, they always add up to 16. Okay? So if you had to go through here and count, you would find that there are 9 that show dominant for the P color and dominant for the P shape, right? That's big Y, big Y, big R, big R, big Y, little r. If you count, that's dominant for the Y and dominant for the R. That's dominant for the Y and dominant for the R. That's dominant for the Y and dominant for the R. That one's dominant for the Y and dominant for the R. Yeah, but what about that little r? Remember, it's recessive. If it's got a big R, it covers over the little, so we count it as dominant. Dominant for both. But look at this one. This one is not dominant for both. This one's dominant for the first one. I'm sorry, recessive for the first one and dominant for the second one. So we count that differently. Okay? Then we have a dominant and dominant. Then we have a recessive dominant. So now I've got 6 dominant dominants, 2 recessive dominants. Keep going. Okay? Uh-oh, look at here. This one is dominant for the first one, recessive for the second one. And so there's 3 of those, right? So I've got 3 that are recessive for the first one, dominant for the second one, 9 that are dominant for both, 3 that are dominant for the first, recessive for the second, and 1 that's recessive for both, or a 9 to 3 to 3 to 1. It doesn't always work that way, depending on the parents. The parents could be different up here, which means you could come up with all kinds of weird ratios. I think there's 8 different possible ratios, but they all end up adding up to 16. And that's one way I teach my kids, is you can tell you got this right you can tell if you got it wrong if it doesn't add up to 16. If it adds up to 16, it doesn't necessarily mean you got it right, but if it don't add up to 16, you've goofed something up, because there's 16 possible outcomes. If these were cows and I were looking at 2 traits from that bull and 2 traits from that breeding heifer, there would be 16 possible outcomes for the offspring. Okay? And then I could start working the odds in my head. What are the odds that it's going to come out with the traits that I want? So this is a dihybrid. Can we go bigger? Sure. We can go with a trihybrid. There would be 64 possible squares. We can go with a quadrihybrid, and there would be, hold on, it's n to the 4th. It's, I don't know, in the hundreds. And that gets crazy. That's when you need a computer for that. Here's a closer look at it. But rather than teaching you how to do this, I'm going to show you a video to show you how to do this. And it's going to be better than listening to me. I would strongly encourage you to go watch those videos. Now this particular question just asks you to do a simple monohybrid cross, and just watch the video on how to do it. It's really easy. Just draw your square and add your parents. It tells you what the parents are. One is big F little f. Yeah, never mind. This is a dihybrid cross. This is the hard Punnett square. But you know what? Go ahead and draw it out. Draw it out and see if you can figure it out. But you know what? I can tell you, I'll give you a little hint. It's just like the last one I showed you, the last slide. And so you can maybe figure it out. And all you have to really do is answer this question. What are the chances it's going to get the red flower and the green seed? See if you can figure it out. I'm curious to see your answers. This was simple Mendelian genetics. It shows us how we can take a look at traits that only have one possible outcome, or 2 possible outcomes. You know, tall or short, fat or skinny, that sort of thing. In humans, that's rare to find. It's hard for us to find some traits that have that. There are a few. One of them would be what we call hitchhiker's thumb, and I'll try to show you that. It's a little oversimplified here, but the regular thumb doesn't have the slight backwards curve to it. They're forcing that back, and you can probably do that with yours. I'm sure you're doing it now, but yours may go back some, but a lot of people, man, it goes way back. That's called a hitchhiker's thumb. That's recessive. Okay, there's 2 possibilities: a regular thumb or a hitchhiker's thumb. Let me see what else. Yeah, I'm just going to show you the Google Images hits on these, because it seems like only beautiful people have widow's peaks, which explains why I don't have one. But a widow's peak, I think, is recessive. And so what it basically comes down to, see that little bump right there where their hair comes down and goes back up? She's got it. Of course, Pretty Boy here's got it. DiCaprio's a little too fat to see it, but there it is. You know, Keanu's got one that's like, wow, you know, whoever this guy is, he's got it. That's recessive, okay? So if they have that, that's a recessive trait. There's two possibilities. There's straight across, and there's the little bump. I'll show you one more, but there's all kinds of these in humans, but most of the traits are not this way. These are what we call monogenic traits. They only have two possibilities. And earlobes is another one. See how this lady's ear comes right down, I'm assuming this is a lady, comes right down and then attaches right there, versus dangly or detached. So it comes down and it makes a little dangle, okay? Earlobes, attached or unattached, are also the result of one allele controlling it, okay? And that's really cool. That's a good example of it, but Mendel had to start figuring out some other things. Most of our traits are what we call polygenetic, meaning that they're controlled by multiple genes. It's not just one spot on one chromosome. A lot of our genes, such as skin color, eye color, hair color, are controlled by several chromosomes, and that's much harder to keep track of when you're looking at using a Punnett square. It's really difficult. So your height, your build, how heavy you are, that sort of thing. A lot of that's genetic and is controlled by more than one gene. And your skin color. I mean, if skin color were monogenetic, there would be two skin colors, dark and light. But since we have a wide range of skin colors, we know that it's caused by way more than one gene. Okay? And genes are like light switches. They're either on or off. Think of if you went into a room that had 50 lights in it and they were all controlled by one switch. How many options do you have? Well, you just have on or off, right? But imagine if you could go into a room with 50 lights and each light had its own switch. You could then change the light levels in the room lots of ways. Well, a lot of our traits are that way. We have multiple switches or multiple genes that give us the possibility of having multiple characteristics: tall, very short, that sort of thing. And it's controlled by many genes. Well, one more thing I want you to know about genes is that in your lifetime they can be turned on or turned off. You know, as you go through puberty, certain genes are turned on. They were off from the moment you were born all the way until you hit puberty, and then they turned on. So they're like a light switch, but now the light switch is on, right? And then the same thing as you get older. My hair, I'm reaching an age where my hair is starting to be less colored as it was. I have red hair, and it's becoming very light. And it's my understanding that eventually it'll just fade to white. That's because the color of my hair is controlled by a protein that's controlled by a gene that was on from basically the moment I was born until right about now. They're starting to shut down, and my hair is getting lighter and lighter, and probably in the next 5 or 10 years I'll be all white-headed. But that's what an epigeneticist does. Epigeneticists study more than just how genes are transmitted. That would be a geneticist. An epigeneticist kind of pays attention to how they work over the years. How do they, over time, how do they turn on or off? Or is it time, or is it something else? We find that there are environmental things that can turn genes on and off too. And so an epigeneticist studies that. It's brand new. I mean, this is one of the newest things in genetics. But an epigeneticist is a great career for somebody that's interested in that. Studying things like tobacco smoke, which has the potential to turn off genes or turn on genes, okay? Other types of pollution. And by the way, tobacco smoke can turn on a gene that can then cause cancer. So that's an example of epigenetics, okay? Looking for epigenetic markers that turn genes on or off. Age, just all kinds of things can affect whether genes are turned on or off throughout the life of the organism. And that's what an epigeneticist studies. It's a very interesting career, one that you may be interested in looking at. That's why we're showing it to you. And that's it. This was a real long one, y'all, and I totally understand. I tried to go easier on you this week, and I'm going to give you a little bit of time next week. But I need you guys to slowly digest this material. It is not easy, and you need to be going through this several times. Definitely watch the videos. Now more than ever, these videos really help you understand a whole lot better what is having to be learned. You need to spend a lot of time this week reviewing this before you take the mastery test. But even more importantly, you need to make sure you have all this in your notes and ready to go, so that when you take that exam, which is coming up very soon, the unit exam is going to cover a lot of this material. So make sure you have this in your head. This is tough stuff. So refocus and spend some time on it. All right, I'm done. Good luck.